we're going to talk about cell transport, and this is how cells get things in and out of them, which is really important because the cell has to maintain homeostasis, so it's constantly moving things in and out like water and waste and food and minerals, so it has to go in and out through that cell membrane, which is the cell organelle that's going to help maintain homeostasis for the cell. So there's two types of cell transport. The first one is called passive transport, and passive transport is going to move molecules from an area of high concentration to an area of low concentration. Okay, so we say this goes down or with the concentration gradient. And this one does not require energy because it's going down the concentration gradient. So there's three types. The first type is called diffusion, and this is usually going to be smaller molecules like oxygen or carbon dioxide that can travel directly through the cell membrane. So if we look at these molecules here, I have a high concentration at the top and a lower concentration at the bottom. So these will move through the cell membrane. More of them will travel this way until they're equal on both sides. And once they're equal on both sides, that's called equilibrium. They will continue to move back and forth, but at a constant rate, so that they stay equal on both sides or stay in equilibrium. The second type is called osmosis, which is the diffusion of water. And again, water is going to move from an area of high concentration of water to an area of low concentration of water. So you can see I have more molecules of water up here, which means I have a higher concentration of water. So these will travel directly through the cell membrane, or they can go through a channel that looks similar to this called an aquaporin that allows water to move in and out of the cell. Okay, so again, they will move from an area of high to low until they're equal on both sides, and that's when they reach equilibrium. They will continue to move back and forth at a constant rate. The last type is called facilitated diffusion, and this is typically going to be larger molecules like glucose that are too big to fit directly through the cell membrane. So instead, they go through a channel in the cell membrane called a protein channel, or a transport protein. So this just allows larger molecules to go through. So with facilitated diffusion, we're still going from an area of high, so you can see we have more up here, high concentration to low concentration, but they're just going through the protein channel. So again, they will go through until they're even on both sides and they reach equilibrium. Now with osmosis, we have another type of solutions called tonicity. And tonicity, we follow the water from high to low. So a lot of times you'll see examples where you have a cell inside of a solution, and inside the cell has to be 100% solution, and outside the cell has to be 100% solution. So whatever number they give you, you just subtract it from 100 to get your other percentage. So if I have 10% salt, that means I'm going to have 90% water. And then outside of the cell, I have a different solution that has 12% salt, which means I'm going to have 88% water. So when I have this, I'm trying to figure out the solution of the outside of the cell here. So I just draw my arrow from the higher number of water to the lower number of water. So high to low. So water is going to move out of the cell. And I kind of think of it as like a water balloon. If I let water out of a cell, it's going to get smaller. And whenever it gets smaller, we call that a hypertonic solution. The second one here, I have a 10% salt solution inside the cell, which again means I have 90% water. Outside the cell, I have 8% salt, which means I have 92% water. So again, water moves from high to low, so I just draw my arrow from the bigger number of water to the smaller number of water. So in this case, water is going to move into the cell. Okay, again, if I put water into a water balloon, it's going to get bigger. And whenever that happens, we call that a hypotonic solution. And the way that I remember that is hypo-hippo. Hippos are really big, so whenever you have a cell in a hypotonic solution, it's going to get really big, like a hippo. And the last type of solution will have actually equal concentrations inside and outside. So inside the cell I have 10% salt, which means I have 90% water. Outside the cell I have 10% salt, which means I have 90% water. So whenever they're equal, again, remember if you think back to up here, once they're in equilibrium, they do still move in and out at a constant rate, but since it's a constant rate, the cell won't change size. So the cell will stay the same size. Okay, so water is going to move in and out at a constant rate, and we call that an isotonic solution. So again, tonicity is the movement of water, so this would be osmosis. And down here, the last part, we have active transport. So active transport is kind of the opposite of passive transport. Typically, it's going to move from an area of low concentration to high concentration. And it does require energy, or ATP. So just like with passive transport, we have 3 types of active transport. The first one is called a protein pump, and it looks very similar to facilitated diffusion. However, it has to go through this protein channel, or a protein pump that's embedded in the cell membrane. So what happens is it opens up on one side of the membrane, the molecules come in, it closes, and then it opens up on the other side so they can get pumped across the membrane. So this one, we're going from low concentration at the top to a high concentration at the bottom, which would tell us it's going to be active. Now, a lot of times you'll also see they'll have like a starburst with ATP being used, and that is a big clue that it's going to be active transport. Anytime you see ATP, you should think active transport. Now, the next type is called endocytosis, and endo is entering the cell. So this is where a whole cell is going to take in molecules. A good example of that would be like a cell eating something else. So what happens is the cell membrane actually kind of pinches in a little bit, so the molecules get folded into the cell membrane. It forms what we call a vesicle, which I like to think of them as like a little transport bubble, around those molecules to bring it inside the cell, and then it will open up to release whatever was brought in to the cell. The opposite of that is exocytosis, so I think exo exiting. Okay, so it looks exactly the opposite. So the molecules will come into this transport bubble. It will transport them to the edge of the cell, and then it will fuse with the cell membrane to release the molecules outside of the cell.